Now, the first experiments on osmosis. We have the next experiments on osmosis. And the first experiment we have Thistle funnel experiment. What we have Thistle funnel experiment. That is the first experiment, Thistle funnel experiment. All right. Now, in this Thistle funnel experiment, I am making the setup here. Beaker containing water. We have the Thistle funnel here. This is beaker. Water. This I am making. This is SPM, semi-permeable membrane. All right. In this funnel, what we have here, sugar or salt solution. Sugar or salt solution. Now, with the help of the clamp stand and all, we are just holding this clamp stand, this for the support of this, holding it the accessible funnel. All right. Now here, how the board has asked you, 10 questions was asked in this Thistle funnel experiment in the board. First, we discuss what is Thistle funnel experiment. Then we will discuss the questions on Thistle funnel. Now, what you have to do in Thistle funnel experiment, take a Thistle funnel, with its mouth covered with semi-permeable membrane. First thing, take a Thistle funnel with its mouth covered with semi-permeable membrane. Got it? And semi-permeable membrane, I have already told you, you go to Ranier bladder, collodion, parchment paper. All right. These are the semi-permeable membrane. And fill some sugar or salt solution inside this. And fill some sugar or salt solution inside the Thistle funnel. And suspend this funnel in a beaker. And suspend this funnel in a beaker containing water. So this is a beaker containing water, and you are suspending Thistle funnel with the help of the clamp. All right. And observe after some time. First thing what we have here, observation. Write it. Observation. Now observation. Water. Hypotonic. The movement of water will take place. Yes or no? And osmosis. Observation. What will happen? The level. Observation, always remember what you see, that is observation. So when the water is moving from here, beaker of water into the Thistle funnel, so observation may you will like the level of sugar or salt solution in the Thistle funnel increases. The level of sugar solution or the salt solution in the funnel increases. Then the second point, what is the reason for observation? Give reason. Reason for observation. Water in the beaker is a hypotonic solution as compared to sugar or salt solution in the Thistle funnel, which is hypertonic solution. So water of the beaker enters into the Thistle funnel by end osmosis through semi-permeable membrane. All right. So this is reason for observation. Now, what will be your conclusion? What you concluded? That water molecules move from hypotonic to hypertonic solution through semi-permeable membrane. or you can write osmosis, simply the osmosis definition. This experiment demonstrated what? Osmosis. Alright, that is a hypotonic to hypertonic solution through semi-permeable membrane. Clear? This is the first experiment, that is Thistle funnel experiment. Now, how the board asks you the question? Here, the first thing, diagram given in an experimental setup to demonstrate a certain biological process. This will be the diagram they will give you, and say diagram given in an experimental setup. This is the experimental setup. Diagram given in an experimental setup to demonstrate certain biological process in plants. Now they ask you 10 questions in ICSC regarding this. This is very important. First one, what is the aim of the experiment? Write down the question. First, aim of the experiment. Second, name the biological process. Third, what will you observe after half an hour of the experiment? Means third thing they ask you regarding observation. Fourth, reason for observation. Fifth, conclusion, which I already told you. Sixth, what will you observe? This is very important, sixth one. What will you observe if the experimental setup is reversed? Seventh, what will we observe if the experimental setup is reversed? Seventh, where, this is applied based question now. Question number 7. Where does this process occurs in nature in plant? Where does this process occurs in nature in plants? Question number 8. Compare the component of this experimental setup. Question number 8. Compare the experimental setup. Compare the components of experimental setup with that in nature. These are the 8 questions which you have to do. All right, so aim of the experiment. What is the aim of the experiment? The aim of the experiment is to demonstrate that water molecules move from hypotonic to hypertonic solution through semi-permeable membrane. Aim of the experiment. Name the biological process. Either you will write osmosis or endosmosis. In both you have the marks. Third, observation. I've already told you. The level of sugar or salt solution in the Thistle funnel increases. Reason for observation, I've already told you. Conclusion, already told you. 6. If the experimental setup is reversed, means in the beaker you have sugar or salt solution and in the Thistle funnel you have water. When the experimental setup is reversed, the level of water in the funnel gradually fall due to X osmosis. Remember that. Due to X osmosis. If the experimental setup is reversed, means here the water and here is the sugar or salt solution. So observation you will write here. The level of the Thistle funnel gradually falls due to X osmosis. Now, Where this occurs in nature? This process occurs in nature in so the question number 7, what was that? Where does this process occurs in nature in plants? In nature in plant in root hairs. In nature in plants in root hairs. All right. Now the last question what we have, compare the component of this experimental setup with that process which occurs in nature. So what you will compare here? Here is the sugar or the salt solution. Here is a thistle funnel, water in the beaker. By making it here, this is a semi-permeable membrane, all right. Now, when you have the root hairs, in the root hairs, what we have this here? Here we have the soil solution. So here, water in the beaker will be treated as a soil solution. They ask you, compare the component of the experimental setup with that in nature. All right. So what will be the water in beaker? We are comparing water in beaker is equal to soil water. Water in beaker is equal to soil water. All right. Then sugar or salt solution. Sugar or salt solution. So this root here, I have already told you. Root here having the vacuole. Vacuole having the salt, sugar or salt solution. So this sugar or salt solution in the thistle funnel will be equal to the cell sap in root will be equal to the cell sap and root here. All right. And this funnel holding all these, so this funnel will be equal to root hairs. The funnel will be equal to the root hairs. Sugar or salt solution that will be equal to the cell sap in root hair, funnel root hair, and water in the beaker is equal to the soil solution. You can see it from here. Soil solution, water in the beaker, this thistle funnel, this SPM, semi-permeable membrane. Clear? So these are the questions that would have asked you regarding thistle funnel experiment. I hope this experiment is very much clear. This experiment as well as the questions which would have asked on this thistle funnel experiment. Again, remember, diagram given in experimental setup to demonstrate a certain biological process in plants, then they ask you all these questions. Now, next experiment we have. First one was the thistle funnel experiment, and the second we have potato. Osmoscope. Potato osmoscope experiment. All right. Take a fresh potato tuber, peel it. All right. And when you are having the fresh potato tuber, a big one, and then scoop out with the help of the spoon or from the knife, scoop out. So, potato osmoscope experiment, take a fresh potato tuber, remove its peel, then scoop out a cavity. Then scoop out a cavity like this. Potato tuber. Now, this is not the circle. I'm making just the circle in of the potato. Let's see. Circular. Then scoop out the cavity like this. This is a cavity. To take a potato tuber and peel it, then scoop out the cavity inside the potato tuber and fill some sugar or salt solution inside this cavity. All right, take a fresh potato tuber and remove its peel and then scoop out cavity inside the potato tuber and fill the cavity with sugar or salt solution inside the cavity. And place this potato tuber in a petri dish or in a beaker, in a petri dish containing water. And to put it in this petri dish containing water. And observe after some time. Again, observation, aim of the experiment, observation, conclusion, procedure. What you will write in this procedure? Take a potato tuber chip or a potato tuber, remove its peel, scoop out its cavity inside the potato tuber and fill the cavity with sugar or salt solution and place this potato tuber in a petri dish containing water or in a beaker containing water and observe after some time. Observation. So what will we see in the observation? The sugar or the salt solution. Sugar or the salt solution level in the cavity rises. Right observation, the level of sugar or salt solution in the cavity of the potato tuber rises. Once you know the observation, you have to give the reason. Yes, petri dish or the beaker containing water is a hypotonic solution as compared to sugar or salt solution in the potato tuber. Therefore, water molecules move from the beaker into the water molecules move from hypotonic solution to hypertonic solution through semi-permeable membrane by end osmosis. You will use the word end osmosis, not only osmosis, end osmosis. So water in the petri dish or water in the beaker is hypotonic solution. Therefore, water molecules move from hypotonic to hypertonic solution through FPM by end osmosis. This results in the rise of sugar or salt solution. Conclusion. The experiment demonstrates that water molecules move from the region of higher concentration to the region from hypotonic solution to hypertonic solution through semi-permeable membrane. Now, again, this potato osmoscope experiment, they ask you the question. Same thing, diagram given in experimental setup to demonstrate the certain biological process in plant. In plant. All right. Or in this potato tuber. One, biological process and osmosis. Observation, aim of the experiment, conclusion, reason for observation. Everything you have to write it. The same way they have asked you the question. All right. Now, you have studied so much osmosis. What is the significance of osmosis in plants? These are the experiments on osmosis. Next, we have significance of osmosis. Importance of osmosis. Significance of osmosis. Importance of osmosis. Number one, you have seen in nature, root hair is absorbing water from the soil by endosmosis. Right, number one, plant absorbs water from the soil by the root hair by endosmosis. Number one, plant absorbs water from the soil by the root hair or by root by osmosis or endosmosis. All right. So absorption of water from the soil by the root or root hair by osmosis or endosmosis. Second, stomata opens in the daytime because of turgidity. We will, when we study the transpiration, then we go through the mechanism of stomatal opening and closing, how that stomata opens. So stomata opens because of endosmosis, because of turgidity. So stomata opens due to endosmosis. The second, osmosis in guard cells. So osmosis you will find in the guard cells also. We will go through that. Osmosis in guard cells results in stomatal opening and closing. Osmosis in guard cells results in stomatal opening and closing. This osmosis develops the turgor pressure. You have seen that osmosis in plant cells, turgor pressure. Osmosis develops turgor pressure in plant tissues and organs become stiff, rigid. So this is very important. Osmosis is providing hardness, that is rigidness to the organs. Then osmosis you see turgidity, and turgidity helps. In class 9 you have studied the radical and plumule. So turgidity helps in growing radical and plumule. Helps in growing radical and plumule. You know, radical give rise to the root system. Plumule give rise to the shoot system. So turgidity helps in growing radical and plumule. Next point, turgor helps in uncoiling of leaves, opening of flowers, and in turgor movements. So these are important significance of osmosis. One more you can add, that is the distribution of water from cell to cell in the entire plant body by osmosis. All right. So this much you do in this class. Next, we will go for plasmolysis, deplasmolysis, and there we have the so many reason-based question. Excess sugar is added to jam and jellies. Excess salt is added to pickle. All right, then the diagram-based questions would ask you, they have made the root here and some alkaline solution is there. What will be the diagram for that root hair, how the root hair turns, and we have imbibition, absorption of water by plant, then the difference between turgid cell, flaccid cell, ascent of sap. This you have to do, and then we'll go through the each and every questions of concise. All right, each and every questions we will discuss of this chapter, absorption of water by roots in this plant physiology. That's it. Thank you.